Hello students. So I'm back again with your second class. In this class, as I've already told you, we are going to start with mechanism of transcription. So let us start first with prokaryotic transcription. Now if you remember the structure of the transcription unit, you already know there would be a promoter, there will be a terminator, and in between will remain the structural gene which contains the code for making the protein, isn't it? So now in prokaryotic transcription, the first thing that I would like to mention is about the enzyme, the main enzyme associated with the process of transcription. In prokaryotes, the main enzyme is only of one type, that is RNA polymerase. Mainly RNA polymerase in prokaryotes can be of only one type, that is a holoenzyme, and it's made up of various polypeptides, namely alpha, beta, beta prime, omega, sigma. Alright? Alpha 2, beta, beta prime, omega, sigma. Alright? Now, other than the sigma factor, this entire enzyme will be termed as core enzyme. Alright? It will be termed as core enzyme. Now, the sigma factor is going to be recognizing a sequence that's present in the promoter site. In the promoter site, there is a specific sequence called the recognition sequence, which is only recognized by the sigma factor. So sigma factor will allow specific binding of the RNA polymerase with the recognition sequence. So it will allow specific binding of the RNA polymerase so the transcription process can begin. Alright. So do remember here one important thing. Without the sigma factor, this core enzyme, it can bind to the DNA and it can also make a RNA, or it can also polymerize a RNA form or RNA strand. But then it will not know where exactly to go and bind. So in that case, without the sigma factor, the core enzyme will be able to make a RNA, but then the transcription process will then start randomly from anywhere. There will be no specific location from where the transcription begins. And of course, that will make the entire process meaningless. Alright? So now, the process, how it occurs. Now suppose this is going to be the transcription unit. This being the promoter, this being the terminator, this being the structural gene. Alright? In case of prokaryotes, there is a specific recognition sequence that's present at -10 base pair of the promoter site. See, structural gene, from where the structural gene begins, we mark that point to be 0. It's the starting. So, if this is the 0 point, before that all the bases can be termed as -1, -2, -3 likewise. So, in that case, for prokaryotes, in the promoter site at -10 base pair, there will be a specific recognition sequence, and that recognition sequence would be called TAAT. This is the sequence. Alright, and this is known as Pribnow box. Pribnow box. There can be another recognition sequence also that may be present in the promoter site, that would be present at -35 base pairs, and this would be called as, or this sequence is going to be TTGACA. That is TT-GACA sequence. Alright, it will be present at -35 base pair. So these are specific recognition sequence where the sigma factor will go and bind, and hence the RNA polymerase's binding will become specific and the process will start. Now, suppose the RNA polymerase have successfully bound to the transcription unit. It now starts moving forward and starts moving towards the structural gene. As soon as it reaches the structural gene, as soon as it starts the structural gene, it will now start reading the template strand, that is 3' to 5'. Okay? And as per base complementarity rule, it will start making one RNA. Now, polymerization or formation of the RNA is going to be exactly like replication process. If suppose there is a A here, then instead of T, here there will be a U. If the next sequence suppose is C, the RNA polymerase will be bringing along a G as a complementary base. So as per base complementarity rule, the entire RNA will be made. So let me show you how it will be made. Suppose this is the sequence here, G, C, T, T, A, G, C. This is the sequence present in the template strand. Now RNA polymerase, here there is an important difference. See, RNA polymerase will be, there is a basic difference here about RNA polymerase and DNA polymerase. RNA polymerase enzyme does not require any primer to start the process, which you have seen in replication, that DNA polymerase enzyme, it may be highly specific or it may be very rapid and very precise, but it cannot start the process. RNA polymerase has no such problem. It can start the process. Fine, no primers will be required. So it brings a activated nucleotide, that is here a U, UTP will be brought here, and as soon as that U is brought, UTP is going to be brought, ribonucleoside triphosphate, it will be converted to monophosphate state. Fine, releasing or breaking 2 phosphate bonds. As soon as 2 phosphate bonds will break, energy will be released because peptide bonds are high energy bonds. With the energy being released, the chain elongation will carry on. Alright, for chain elongation, the energy is coming exactly like it had come in case of DNA replication process. Is it clear, all of you? So likewise, as per base complementarity rule, if here there is a U, A, here it will be a U, C, so here there will be a G, likewise G, so C, C, so G, T, so A, again A, likewise A, so U, then there will be C and G. Likewise, the whole RNA will be formed, alright? The whole RNA will be formed as per base complementarity rule. Now see, while the RNA is being formed, do remember, the RNA will keep hanging with along with, it will remain entangled or it will remain bound to the RNA polymerase enzyme. Because it's a DNA strand and this being a RNA, hybridization between DNA and RNA of course won't be taking place. So in that case, throughout the process, the RNA strand that has been formed, it will remain bound to the RNA polymerase enzyme. And because there is no hydrogen bonding with the DNA here, that's why the RNA that is being formed will be hanging and trailing outside the transcription unit, the transcription initiation complex. From here, it will be completely as if it is hanging outside the complex. Likewise, it will be trailing outside. Alright. Finally, as the RNA polymerase will reach the terminator site, there is a particular factor that will be required, that's called rho factor. What factor is it? It's called rho factor. Okay, rho factor will be activated as soon as the RNA polymerase reaches the termination site, and as soon as the rho factor gets activated, it will hydrolyze and break the bond between the RNA and the RNA polymerase, so that the RNA is going to be free. It will come out of the transcription complex. Likewise, the RNA polymerase will also be free so that it will move out, and the part of the DNA that had opened up to allow transcription, that will also start to become coiled up again. Okay, it will coil up again. So this is the process by which prokaryotic transcription takes place. Let me repeat it in brief once again with you so that you can understand it more properly. First of all, during prokaryotic transcription, in the promoter site, mostly at -10 base pair, there will be a specific sequence called TATAAT. This sequence is also called Pribnow box, where the sigma factor will go and bind and the process will start. The RNA polymerase will start moving towards the structural gene, and by base complementarity rule, it will make the entire RNA. Fine? As it makes the RNA, the energy will be coming from breaking down of the phosphate bonds present in the ribonucleoside triphosphates. Now, finally, by base complementarity rule, as the RNA strand is made, completed, and the RNA polymerase reaches the terminator site, and immediately rho factor will be activated and it will stop the process of transcription. Alright? Clear all of you? Fine. Now, let me mark with you, there are major 3 differences between prokaryotic and eukaryotic transcription. The major 3 differences are, first of all, unlike in case of eukaryotes, in prokaryotic transcription, after the RNA is formed, there is no further processing required. So in simple words, in prokaryotes, after transcription, post-transcriptional modification is not required. Alright? It won't be required because the prokaryotes don't possess split gene arrangement. They don't have intron-exon kind of a gene-like arrangement. So in that case, there is no essentiality for any processing. Second point is that in case of prokaryotes, the transcription and translation processes both are occurring at the same place because there is no nuclear membrane in the prokaryotes, so we will not be, it is not going to be occurring like eukaryotes, in which we see that transcription occurs in the nucleus and the translation process occurs in the cytoplasm. No such division is going to be happening in prokaryotes. Both transcription and translation occurs at the same site. And the third point is that in prokaryotes, sometimes it can be seen that much before the transcription process is complete, because you know the RNA here is trailing outside, isn't it? So much before the RNA polymerase could reach the termination site, already a ribosome can bind with the RNA and it can already start translation. So in case of prokaryotes, transcription and translation being occurring at the same place, both are coupled events. Both can be occurring simultaneously, which is not at all a possibility in case of eukaryotes because transcription and translation are occurring at different sites. Is that clear? All of you? Fine. So now, next we'll be discussing eukaryotic transcription. Before we start the mechanism of eukaryotic transcription, you must first know that unlike prokaryotes, in eukaryotes, RNA polymerase can be of 3 types. Okay? First, RNA polymerase 1. RNA polymerase 1 is mainly known for formation of rRNA. Ribosomal RNA, it produces or it forms 5.8S, 18S and 28S rRNA. Mark here, 5S rRNA is not formed by RNA polymerase 1. RNA polymerase 2. It is going to be responsible for the formation of hnRNA, which after processing becomes mRNA. So in the mechanism mainly we will be considering RNA polymerase II. And finally RNA polymerase III. RNA polymerase III is going to be responsible for formation of tRNA. And of course 5S rRNA. And also snRNA, small nuclear RNA, scRNA, small conditional RNA. Alright? So these are the three important types of RNA polymerase found in eukaryotes. But because we are mainly observing or trying to study mRNA formation, that's why we will be considering here RNA polymerase II. Alright? So let's see the mechanism. The mechanism is very similar to the prokaryotes with minute differences. The transcription unit in eukaryotes is also going to be again having three distinct regions, that is promoter, terminator and structural gene. Now, the promoter site in case of eukaryotes also possesses a recognition sequence. This recognition sequence will be present at 20 base pair, and it would be a 7 base pair long sequence that's called TA, 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 T, or TA, T, AA, AT. Alright? This particular sequence is going to be termed as Hogness box. Hogness box is going to be present at -20 base pair of the promoter site. Alright? So there the RNA polymerase II would bind and it would start moving towards the structural gene. As soon as it reaches the structural gene, by base complementarity rule, the entire RNA will be made. The RNA as usual will remain bound to the RNA polymerase enzyme, and it will also be trailing outside the transcription unit. Alright? Exactly same, no difference. Finally, as the RNA polymerase will reach the terminator, there the process of transcription will stop. Alright? So this is the very simple mechanism of eukaryotic transcription. Alright? Very similar to the prokaryotes. It goes and the RNA polymerase II will go and bind to a specific recognition site present in the promoter. Then it will start moving towards the structural gene. As it reaches the structural gene, by base complementarity rule, it will make the RNA transcript. And finally, when the RNA polymerase will reach the terminator site, the process of transcription will complete. Alright? Now, the thing is that the major difference is that here the RNA that is being made will not call it mRNA immediately. Because this RNA that has been formed, it consists of introns that is going to be like, interrupting the exons. So because it is going to be possessing introns and exons, that means there are some unnecessary parts also present in this particular RNA. We do not directly call it mRNA. We'll rather now call it primary transcript, or we will call it hnRNA. Okay? Hn means heterogeneous nuclear RNA. Okay? We'll not directly call it mRNA, because whenever the hnRNA will undergo processing and it will become fully functional, only then it can be called as mRNA. Okay? So let's see how we can convert the hnRNA or the primary transcript into the mRNA. For that, the process that's required is called post-transcriptional modification. So let's see what is post-transcriptional modification. In post-transcriptional modification, certain kind of processing would be done in the newly formed RNA. Alright? So suppose this is the newly formed RNA 3' to 5', 5' to 3' end. In the 5' end, a very unusual nucleotide that's called methyl guanosine triphosphate that has to be added. This adding of methyl guanosine triphosphate in the 5' end is known as capping. And capping is going to be catalyzed by the enzyme guanyl transferase. Alright? Capping is very important a step. Why? Because when the RNA or the mRNA will reach the cytoplasm for translation, the ribosome would have to recognize the RNA and has to come and bind to the RNA. For recognition, this gap is very, very essential. Without the gap, the ribosome will not be able to recognize the mRNA and of course then cannot come and bind with it. Finally, in the 3' end, certain 200 to 300 adenylate residues has to be added. This particular step is known as tailing. And tailing is done or catalyzed by the enzyme poly-A polymerase. Now finally, after capping and tailing, what is left is that certain parts or sequences in this portion of the RNA are unnecessary or are introns. These introns are to be removed. Now removing of intron is known as splicing. Okay. Now for splicing process, there are certain kind of RNA, that is snRNA, along with certain protein complexes called snRNP, small nuclear ribonucleoprotein. These are going to form a complex called spliceosomes, which are going to bind to these introns, and from both ends they are going to simply press the introns in a manner that they will loop out like this. They will simply form loops like this. When they are being pressured from both ends, they are simply going to form a loop. As they form the loop, these loops are to be then removed. They are to be cut. And as they are cut by the spliceosomes, they are going to fall off and separate from the exons. Later on, these exons, they are going to be joined together by the help of the enzyme ligase. Alright. So now, after capping and tailing and splicing, what we get is a functional mRNA. Now we can finally call it mRNA. So this is the transcription process in eukaryotes as well. So let us meet in the next class with the next topic, that is genetic code. Alright. Thank you.